All right. Hello and welcome to my final project/presentation for Biology 4130, Conservation Genetics. I'm Ben Parent, and I hope you enjoy my presentation about Alaris fulgens, the red panda. Okay, so why I selected the red panda? So there were some very serious scientific reasons and very course-related topics as to why I selected this species, and it definitely was not because I find them really stinking cute. Okay, so now that I have the silly shenanigans out of the way, I'm going to get serious and I want to introduce the species. The red panda is an arboreal herbivorous mammal. I had to Google what arboreal meant, and it means that they dwell in trees, so they like to climb, similar to a sloth or something like that. Yeah, so red pandas live in understories of bamboo, that's their main food source, in temperate conifer forests, commonly at high altitudes. I believe their altitude range is from 1,500 meters to 4,800 meters, so that's very high. The red panda is a member of the class Carnivora, but primarily consumes bamboo, which is very strange for a carnivora animal. But they do eat a small amount of meat, so that stays in line with the carnivora distinction. Yeah, so the red panda is currently found in 5 Asian countries: Nepal, India, Bhutan, Myanmar, and China. And the current wild population estimates for the red panda are between 2,500 and 10,000 individuals. So that is obviously a little bit low and a bit concerning. And then I have a picture here. It's a map of the area that the red panda is found, and you can see all those countries listed there. Yeah. So the next item to check off the species introduction list is species appearance. So as you can see in the photos, they look like cute red raccoons, but that's not a very professional description. So here's a more professional one. They're 3.7 to 6.2 kilograms. Their body length ranges from typically from 56 to 62 centimeters. I'm sure there would be some outliers in all these measurements, but these are the general ones that I found. And for my American friends, that's 22 to 24 inches. The tail is an additional 28 to 49 centimeters. The Himalayan red panda has a deeper red coat with more white on its face, and the Chinese red panda is more dark in the coat with pronounced tail stripes. And spoiler alert for later on in the presentation, these two distinctions, these two populations, the Himalayan and Chinese population, were thought to be subspecies of the same species, but in recent research, they were genetically proven or shown to be separate species, so that goes in line with our course topics quite well. Okay, so we're heading into the second subsection of the final project/presentation based on the rubric that I was given, why Alaris fulgens is endangered. So the red panda was classified as endangered in 2015 by the IUCN. Wild population estimates, as I previously stated, are between 2,500 and 10,000 individuals. Red panda populations have been hit especially hard over the past 3 generations, which is 18 years. So if we do some quick math there, each generation is 6 years. Woohoo. The population is expected to decline further if proper intervention is not taken, but through the work of conservation, intervention is being taken, and we will learn about that later. And then population decline is due to a combination of stochastic and deterministic factors, which we learned about in class and are very useful to know about, and I will get into those. If I can shift your attention to the graph on the left, you can see the 20-year decline, so that encompasses the past 3 generations. And yeah, it's not good. If this is your stock portfolio or something like that, you are not happy with those trends. So deterministic factors causing population decline. So as we learned in class, deterministic factors are factors such as habitat destruction, overexploitation of the species itself, pollution, invasive and non-native species introduction. And the more of these deterministic factors that come into play at any one time, the greater the pressure will be on the species and the greater the impact of the stochastic factors that we will see now on the next slide. So yeah, the deterministic factors that are negatively impacting red pandas are habitat destruction. So as I previously stated, red pandas rely heavily on temperate conifer forests for their habitat and bamboo. And there's a big issue going on in their distribution range of deforestation, and a large percentage of it is for cattle farming. So cattle are obviously going to take up a lot of room and graze the fields and all of that stuff. So that's very detrimental for the red panda habitat. And then also the forestry industry of logging, where they're going to be harvesting wood. So that also destroys the red panda habitat. And then poaching is another one of those deterministic factors that we learned about that is negatively impacting the red panda. It's especially bad in China. Um, yeah, their fiery red coat is very appealing for different styles and clothing brands and things like that. So yeah, I will proceed to stochastic factors now. Okay, so we are now focusing on the stochastic factors that are causing population decline. And as we learned in class, some examples of stochastic factors are demographic stochasticity, natural fluctuations in birth and death rates and sex ratios. There's environmental stochasticity, which is rainfall, temperature, food availability, density of predators, etc. Genetic stochasticity, which is loss of alleles, enhancement of deleterious alleles, and random genetic drift. And then there's catastrophes like severe winters, volcanic activity, hurricanes, and disease epidemics. So the two main stochastic factors that are currently acting upon the red panda populations and causing decline are climate change and inbreeding depression. So yeah, climate change is an obvious one here. As the global temperatures rise and forest fires and storms and things like that become more prevalent, it's not going to be good for animals that live in forests or around trees that can burn. And then inbreeding depression is not a huge issue for the red pandas yet. It's more prevalent in the Himalayan populations that have less gene diversity than the Chinese populations. So I'll get into that later on. But yeah, so as the population declines due to the deterministic factors, obviously, if deleterious alleles start to become prevalent in a population and your population numbers are low, then that allele can spread very quickly due to genetic drift in gene flow and things like that. Okay, so we're now heading into the third subtopic of the final presentation slash project, and that's concerning the genetics of the species. So I'm actually going to explain the Alaris fulgens genetics, and then later on the project, I will be explaining the Alaris styani genetics, which is the Chinese red panda. So fulgens is the Himalayan population. But yeah, obtaining genetic information from wild red pandas is extremely difficult due to their nocturnal habits and shyness. And as you can imagine, their shyness is likely due to evolutionary um, factors, such as their being small and cute and cuddly and not so ferocious, so needing to be very shy when it comes to um, interacting with other species and other animals who could be predators. But yeah, so an article that I found sampled 14 loci from 15 Himalayan red pandas. They are being kept in captivity, and this was done at the Padmaja Naidu Himalayan Zoological Park. I'm impressed that I said that correctly, or I might have not, but that's okay. And yeah, I'll be referring to that as the PNHZP for some simplicity, and that's found in Darjeeling, India. And if you look on the right there, you can see a map of India, and Darjeeling is very far northeast, up in the red panda distribution range. And there's a nice sign there, and you can see your little cute red panda hanging out on his branch. So the first genetic concept that I want to discuss is heterozygosity. This was a very important topic in our course notes and the textbook, and it's very important to understand this and to have knowledge of this working in conservation genetics or just any kind of genetics in general. But yeah, heterozygosity, as defined by our notes, is an index of gene diversity. It indicates the expected number of heterozygotes under Hardy-Weinberg equilibrium conditions, and it does not define the observed number. So yeah, you would have to calculate your allele frequencies, and as you can see up top there, you can have dominant and recessive alleles. And then once you have your frequencies, you can figure out the proportions of homozygous dominant, homozygous recessive, and heterozygous that you would expect if the population is under Hardy-Weinberg conditions. And yeah, at the PNHZP reserve, based on their calculations, the heterozygosity should be 0.714. So that means 71.4% of the loci should be heterozygous. And then the observed heterozygosity, so the actual heterozygosity that they measured based on their sample, was 0.681. And this is a pretty small gap, so it's not too much to worry about. And it could be due to things such as sampling error or any kind of variance that occurs when you use sampling. And especially a sample size of 15 is not an overly large sample size. Yeah, so the researchers determined that the population of red pandas there, Himalayan red pandas that is in Hardy-Weinberg equilibrium, and there's no selection forces acting on them. And so, yeah, that's a good indication of pretty good gene diversity levels. Inbreeding is the next genetic topic that was prevalent in this article from the PNHZP reserve. An inbreeding coefficient, as we learned in this course, represents the probability that two alleles are identical by descent. So the probability that the alleles have descended from related individuals and are identical and are homozygous. And a lot of the time, inbreeding is a concern because they can be homozygous, and these negative deleterious alleles can persist in a population because you're not outbreeding to gain diversity. So yeah, in class we did coefficients as fractions or decimal numbers, but this article provided a percentage, so you just multiply the decimal by 100 to get your percent. Easy math. And yeah, so the percentage in the PNHZP population was 4.8% for the inbreeding coefficient. So this is low, and that's a good thing, obviously, because you don't want a high inbreeding percentage. Shout out to Alabama. I'm not sure how Alabama got that stereotype. It's not a nice one, but yeah, that's what comes to mind. But yeah, smaller populations of individuals are much more vulnerable to inbreeding depression, and we can probably figure that out with some common sense. If there's not as many mates available and a small number of couples are available, then whenever they have progeny, there's a high likelihood that you are going to be faced with the only options of related people because the population is so small. I guess not people, any kind of organism. And at the PNHP reserve, they avoid inbreeding through the introduction of new Himalayan red pandas from either other reserves or the wild to come into their reserve and mate and reproduce. And then they also allow some of their red pandas to travel and mate with other populations in order to increase gene flow and make sure that there's outbreeding going on and genetic diversity stays relatively high and inbreeding stays low, because that's what you want. Okay, so the next genetic topic that I want to discuss is genetically viable population factors. This was an important section of the notes and the class, and it's very important to understand. So the notes state that a shortage of habitat is a key reason why many species are threatened or endangered. This definitely rings true for the red panda. As we saw earlier, a deterministic factor causing population decline in the red panda is habitat loss. It's caused by cattle farming and deforestation. And we know that as the population numbers decline, that inbreeding becomes more prevalent. And the notes also state that inbreeding will increase at a rate of 1/2 multiplied by the effective population size. Effective population size is not the same as the actual population size. It is the size of an idealized population that would have the same effect of random sampling on gene frequency as that in the actual population. So effective population size is smaller than your actual population size, and to maintain your desired NE, then your actual population size has to be even bigger than the NE. And I could not find the exact measurement of the red panda effective population size in the articles that I had access to, but I see in the notes that the IUCN states that an effective population size of 1,000 is vulnerable, 250 is endangered, and 50 is critically endangered. So since the red panda population was declared endangered in 2015, I would assume that most red panda populations have an NE of 250 or less. That's a complete inference, but it does add up if the IUCN abides by these criteria. And then another important topic that is in the genetically viable population factors is the MVP. And no, this does not stand for the most valuable player. It is the minimal viable population size, and it is the minimum size required to retain reproductive fitness and evolutionary potential over thousands of years. And I would say it's safe to assume that the red panda is well below its MVP right now because it's facing problems and endangerment. And if it were not below the MVP, then it would not be facing these problems. Now I will be discussing the next genetic topic that I want to discuss that is applicable to the course and the Red Panda project that I'm currently recording. It's taxonomy and evolutionary history. So there's that silly memory trick that we know. King Philip came over for good soup. Kingdom, phylum, class, order, family, genus, species. And yeah, the red panda's kingdom is Animalia, the phylum is Chordata, class Mammalia, order Carnivora, family Ailuridae, genus Ailurus, and then there's two species that were recently discovered that they were not subspecies. They're two separate species. There's Ailurus fulgens, the Himalayan, and Ailurus diane, the Chinese. Okay, so taxonomy and evolutionary history at first glance may not seem like a super important genetic topic, but as we learned in class, it is. The class notes state that taxonomic status must be accurately established so that endangered species are not denied protection nor effort wasted on abundant species. Genetic information assists in resolving taxonomic uncertainties and defining management units within species. And this actually is very applicable to the red panda, as recently two subspecies were correctly identified as actually separate species, the Himalayan and Chinese. I'll talk more about that later. The notes state that errors in the assignment of taxonomic status can have many consequences. Most of these are bad consequences. Unrecognized endangered species may be allowed to become extinct. Endangered species may be denied legal protection, while populations of common species or hybrids between species may be granted protection. Incorrectly diagnosed species may be hybridized with other species, resulting in reduced reproductive fitness. Resources may be wasted on abundant species of hybrid populations, and populations that could be used to improve the fitness of inbred populations may be overlooked. So it's very important to have accurate taxonomy and to understand the evolutionary history of species. So as far as the red panda evolutionary history goes, 30 to 50 million years ago, that's a long time, the taxonomic family Ursidae, which is the modern bears, split into two distinct lineages. One of the emerging groups was the Procyonidae. I apologize if I'm not pronouncing that correctly, but that's how I believe it's pronounced. And then shortly after, the Procyonidae radiated into the New World Procyonids and the Old World Procyonids. The New World Procyonids are the beloved raccoons who jump in our trash cans and ruin things. And then the Old World Procyonids are the lesser panda, which is also known as the red panda. Giant pandas and red pandas are not as closely related as their names would indicate. The giant panda is a member of Ursidae, while the red panda is a member of Procyonidae. Yes, so I'm going to elaborate further on why the red pandas and giant pandas are imposters and that their name infers that they're more closely related than they actually are. They are named similarly because of their convergent evolution. Convergent evolution is when traits evolve simply due to evolution and similar pressures rather than homology. So it is not. So they did not evolve similarly due to a relationship that is hereditary. It's similar evolutionary pressures. So they both eat bamboo, and they both have similar appearances. Yeah. So the first true red panda is believed to have originated in Spain during the Miocene era, which is approximately 5 to 20 million years ago. Okay, so the next genetic topic from the course that pertains to the red panda very well is genetic distance. The notes state that genetic distance is a key tool in discerning whether allopatric populations are subspecies or separate species. This is really applicable to the red pandas because recent research on red pandas has identified two separate species of red pandas that were believed to be subspecies in the past. So in previous research, the Himalayan red panda and Chinese red panda were thought to only have significant differences in morphological appearance, but this was changed in a new research article that I have a quote from here. It states that the haplotype networks and phylogenetic trees of both 8 Y chromosomes, SMP, YSMP haplotypes from 49 male individuals and 41 mitochondrial genome haplotypes from 49 individuals showed that the MH haplotypes from the Himalayan red panda clustered together and separated from the haplotypes of the Chinese red panda. So obviously, if the MH haplotypes from the Himalayan red panda are associating with each other and not associating with the Chinese red panda haplotypes, then there's obviously a large genetic difference that is significant and shows that these two species are not only morphologically different, but also genetically different as well. Okay, so now that we have established that there are two separate species of red pandas, Ailurus fulgens, the Himalayan, and Ailurus diane, the Chinese, I want to talk about the Chinese red panda genetics and give them the attention that they deserve. So, an article that I found concerning the Chinese red panda sequenced a 236 base pair fragment of the mitochondrial D loop region of 53 Chinese red pandas. I actually went back in this article and read more closely, and there were 5 Himalayan red pandas and 48 Chinese. This was an article that was from 2001, so they hadn't completely identified that these two species are separate species, and so this represents the Chinese red panda quite well, but it's not exactly perfect because 5 out of the 53 are Himalayan. But anyway, the researchers found that there are 17 variant sites out of the 236 base pairs that they sequenced, and there were 25 total haplotypes with the sample of 53. And a haplotype is a set of DNA variations or polymorphisms that tend to be inherited together. It can refer to a combination of alleles or to a set of single nucleotide polymorphisms found on the same chromosome. So since there were 25 haplotypes within the sample of 53, which is quite good, the genetic diversity was found to be 0.93 plus or minus 0.02, range of error there. So this is a good genetic diversity, and it indicates a diverse genome. So it is evident that the genetic factors are not the root cause of Chinese panda endangerment because their genetic diversity is quite good, and it's likely the deterministic factors. Okay, so to elaborate on the genetic distance portion of this project, I just want to state an important finding that researchers found. So yeah, this knowledge concerning the genetic diversity between Himalayan red pandas and the Chinese red pandas has researchers excited about potential conservation implications. I have a quote from a recent research paper here where they discovered that the distribution boundary that they believed was the boundary in the past is not actually the right boundary. But anyway, for a long time, the unclear status of species classification and distribution boundary hindered the scientific design of conservation measures. Because of the wrong distribution boundary, the EH-GLH population was split to belong to two species, which could result in inappropriate conservation measures for EH-GLH population and possibly detrimental interbreeding between the two species in captivity. So researchers previously thought that the Nujiang River was the distribution boundary, but is actually the Yalu Zangbu River. So it's very important for scientists and conservation geneticists to know this because they can now correctly identify the range of the Himalayan and the Chinese red panda when they want to breed. So if they want to interbreed them, then they'll know where to release certain pandas to come in contact with certain populations, and if they want to outbreed, then they also know this information. And you can see on the chart on the right there where the Yalu Zangbu River is. There's an arrow pointing to it. And then you can see where the Nujiang River is. So there's a pretty big difference between the location of those two rivers. All right, so this is my final genetic distance slide, and I just want to share a quote from the research paper that identified the correct distribution range for the Himalayan and Chinese red pandas, and also identified that they have significant genetic differentiations and not just morphological ones. But anyway, the Himalayan red panda was estimated to have a small population size, and thus maintaining and increasing this species' population size and genetic diversity are critical for their long-term persistence. In particular, the Himalayan red panda population spans southern Tibet of China, Nepal, India, and Bhutan, which needs urgent transboundary international cooperation to protect this decreasing species. So now that they've correctly identified the distribution range and they've identified the genetic differences and that the Himalayan red panda's genetic diversity in population and effective population is smaller than that of the Chinese red panda effective population, and now they can focus their conservation efforts on the Himalayan red panda more so than the Chinese red panda and try to save this species, Alaris fulgens. We are now moving into the fourth and final subtopic of this final presentation slash project, which is conservation efforts. This is a much happier subtopic because it's some positive things that are being done to help save the red panda as opposed to explaining why the red panda is endangered and in trouble. But anyway, so as we learned in class, diagnosis of genetic problems is crucial for proper conservation efforts. And as I've been talking about the recent research done to identify the Yalu Zangbu River as the actual geographical boundary between the Chinese and Himalayan red panda was crucial because now conservation geneticists can focus on the Himalayan red panda range more so than the Chinese red panda range because the Himalayans need more attention and more help. And these same researchers also uncovered important information about genetic diversity and previous bottlenecks that occurred in each of these red panda species, and I'll talk about those now. So the researchers found that Alaris fulgens, the Himalayan red panda, has undergone 3 population bottlenecks and 1 very small expansion. This has caused genetic diversity to be low, linkage disequilibrium to be high, and genetic load to be high. Whereas in contrast, Alaris styani, the Chinese red panda, has only experienced 2 bottlenecks and 1 large expansion, making genetic diversity better than that of the Himalayan red panda. So this led researchers to the conclusion that the Himalayan red panda requires more attention for conservation efforts due to their lower genetic diversity. And if you look over on the right there, you can see a good example of how population bottlenecks cause lower genetic diversity, because you have a bottle where you have purple, green, and yellow balls or alleles. And when the bottleneck event happens due to variance, you can lose some of those alleles and only get purple. And then your surviving population has less gene diversity. This has happened to the Himalayan red panda 3 times and the Chinese red panda twice, so it makes sense that the Himalayan red panda would have lower diversity than the Chinese. So in terms of actual conservation efforts that are put in place and happening right now to save red panda populations, there have been many reserves established in the red panda distribution range. 46 reserves in China, 20 in India, 7 in Nepal, and 5 in Bhutan. The PNHZP that we discussed earlier in Darjeeling, India, is an example of genetic augmentation effort that is taking place. The PNHZP introduces a new male and new female into the reserve for breeding every 1 to 2 years for genetic augmentation, and will release some of their captive red pandas back into the wild to promote gene flow and genetic diversity. So that's another example of genetic augmentation there, releasing their red pandas back into the wild. And things like this help very much to maintain genetic diversity and gene flow and to strengthen the red panda populations. Okay, so I want to conclude with some conclusions, because that seems like the logical thing to do. Based upon research that I have collected and read through, examining both the Himalayan and Chinese red panda, genetic factors are definitely not to blame for species endangerment. Even though the Himalayan red panda's genetic diversity is weaker than that of the Chinese red panda, it's still not that weak. As we saw at the PNHCP reserve, the inbreeding coefficient percentage for the Himalayan red pandas that were captive there was still only 4.8%, which is really low. And their heterozygosity, their observed heterozygosity was 0.681, which is quite good. So I definitely wouldn't say that the genetic factors are causing the red panda trouble. It is definitely the deterministic factors caused by humans. Humans are not very mindful in certain scenarios, and deforestation and cattle farming so that we can have cheap beef and yummy burgers at McDonald's is really not too sensible and friendly to our friendly critters like the red panda. Um, Here's a quote from Kumar et al. Talking about the Darjeeling Reserve that is very hopeful for the future and shows that there is some hope for the red panda and that we can save it. Being located in the species range, Darjeeling and Gangtok's captive red panda populations have tremendous potential to grow, maintain the genetic diversity of the international captive population, and restock dwindling wild populations. And I certainly hope that Kamar is correct in saying that, and that the red panda is saved, because they are very cute and very nice. And I would like to say thank you to Dr. Hale for a wonderful semester, even though you were feeling a bit under the weather the whole time. I feel like we still learned quite a bit from this course and got some value out of it. And this is my final presentation as a biology student at UPEI, so it's a little emotional, and it's just great to have had a good semester, even though all this COVID stuff's been happening. And I want to thank you again. Okay, bye bye.